to go into these culverts to do this with us. So specifically thanks to Bat Conservation International, Amanda Adams, Sarah Stankovich, as well as all of the undergraduate and graduate students from my university have volunteered their time, as well as Ash Cable, who came from University of Tennessee to essentially train some of our students in how to attach some of the equipment that we used. So this project, again, would not have happened without these individuals. So I'm going to get started in talking about the epidemiological triangle. So essentially, this is the three things that have to happen for a disease to occur. We have to look at a host and the host physiology and behavior that makes this host susceptible to a particular disease. The pathogen, how the pathogen dynamics work, how it's transferred between individuals, how does it grow? And then the shared environment. And someone like myself who is very much interested in physiology and thermobiology and how external environmental conditions such as temperature influence things like animal behavior and animal processes, something like this is really something that brings me into disease ecology, even though I don't consider myself really a disease ecologist. And we're specifically interested in that shared environment when we're talking about particular pathogens that are caused by ectoparasites. So we tend to think about ectoparasites as things like ticks, lice, scabies, mites, etc. But we can also look at ectoparasites as things like fungus. So these are particular parasites that exist on the outside of a host body. So that external environment is even more important to look at because they're not going to be within a host body and be protected by that host body, but rather exposed to those environmental conditions where the host is actually living. And so from a physiological standpoint, this is something that's really interesting to me because we're not just looking at how things like temperature and humidity can influence the organism, such as the host, but also how does it influence these disease dynamics. And to give you some context, particular fungal diseases that we see in wildlife are things like chytrid fungus, which has impacted about 350 amphibian species worldwide. It's waterborne and it survives in the same environments that amphibians do. We also have snake fungal disease. This causes snake behavioral change throughout the eastern US, and it can persist in substrate and can infect snakes by through contact with the environmental reservoir. And then we have what I'm looking at, and that's white nose syndrome. So white nose syndrome is caused by the fungus Pseudogymnastus destructans, and it is one of the most devastating wildlife diseases that we have seen in our lifetime and has killed millions and millions of hibernating bats since we first discovered it in 2006. So coming from that shared environment perspective, we can look at this fungus that causes white nose syndrome and why it's so prolific for these bats. So white nose, the fungus can persist in environments that are fairly cold for a fungus. We usually think about fungi living in moist and warmer conditions, but this particular fungus does really well in cooler conditions. So it can survive between 0 and 19 degrees Celsius, but it thrives extremely well between 12.5 and 15.8 degrees Celsius, which, if you've ever gone into a cave, is fairly conducive to these caves that bats hibernate in. So essentially, we call any place that a bat is going to be hibernating in as hibernacula. And hibernacula essentially will have very similar conditions to where where this fungus can grow extremely well. Uh-oh. Sorry. Okay. So before we start talking about our research, I need to give you a little bit of a crash course in hibernation ecology, specifically for bats. So unlike what we think about hibernation, hibernation is not a steady state. It's also not sleeping. So these animals are not sleeping when they go into hibernation. Rather, this is a reduced physiological state where an animal, such as a bat, will drop its metabolism and drop its body temperature to fairly low levels. And when it's doing this, it usually does this in a time period of energetic constraints. So 42 of the 47 bat species that we have in the United States are insectivores. Insects don't usually survive below freezing. So if you have bats that live in areas like the Northeast where you get snow and you get temperatures below freezing, they don't have any food resources during those winter months. So to reduce the amount of energy they need to expend, they'll go into hibernation and they'll stock up on fat stores before going into hibernation and just be able to persist throughout the winter without having to consume more calories. So during hibernation, while they drop their metabolism and body temperature down, this is called torpor. But again, this is not a steady state. So as you can see in these images, and I'm going to see if I can use my little pointer here. Maybe, okay, it's not letting me. Ah! Okay, sorry about that. I was trying to point to this figure here, but essentially you can see in the top right figure, we have time on the x-axis and you have body temperature on the y-axis. You have this bat that's going in and out of torpor, and we call those peaks of temperature and that peak of metabolism an arousal cycle. And why these hibernators go into arousals is still actually argued about. But what is important to know that they do go into these arousal cycles. And when an animal has to bring its metabolism and body temperature back up to normal, this is really energetically costly. So you can see how a bat that, say its body temperature is around 10 degrees, it has to increase its whole body back up to normal body temperature around 38 or 39 degrees Celsius. And that can cost a lot of energy. And so even though the amount of time that hibernators are in arousals is about 5% of the entire winter period, these arousal periods expends up to 95% of that winter energy. So this is if you blew your monthly budget in 5% of the time, very, very quickly. And so this is how white nose syndrome actually causes mortality in these animals. As you can see in the top two figures, this is, again, a temperature trace over time where the bat's skin temperature is going up into those very short arousals and then coming back down into torpor. And usually when they're in torpor, they're matching air temperature because it reduces how much energy they need to keep that body temperature going, because they're just essentially thermal neutral with their environment. But as you can see in the bottom two figures, those arousals start to increase. And this is because the fungus that causes white nose syndrome, as you can see in this photo of this bat, is going to start to eat away at the wing tissue, which causes that bat to get dehydrated, amongst other things, and it has to come out of torpor. So that increased number of arousals actually causes them to expend all those fat resources before the end of winter. And essentially they starve to death because there's not a lot of food for them to replenish those energy stores. So we've seen white nose syndrome across the United States at this point. It was first discovered, the fungus was first discovered in a cave outside of Albany, New York in 2006. And since then it has spread northward, southward, and westward. It's important to note that we do find the fungus in some areas where we don't actually see the symptoms of white nose syndrome. What's also really interesting to note, as you can see, there is a southern edge of this distribution of this fungus. And this is because the fungus and where white nose syndrome occurs is limited by the hibernation behavior of bats. So bats in Florida may not go into hibernation, and therefore we're not going to see white nose syndrome because these bats are active all year round. That fungus can't grow at skin temperatures that are outside of that zone of that fungus. And so we can look at some really neat physiology, disease, and behavior along that southern range of that fungus. And that's basically what was the start of this particular study, looking at hibernation behavior and fungal dynamics in Texas. And we were particularly interested in looking at the tricolored bat. The tricolored bat is one of the smallest bats we have in North America. It's about 5 to 7 grams, so it's about the weight of a quarter, which is pretty small. Because of this, they don't have a lot of capacity to hold fat. They are one of the hardest hit by white nose syndrome. And we actually just recently petitioned these bats to be listed as an endangered species. And it was the first bat to be petitioned solely on declines from white nose syndrome compared to other things such as deforestation, agricultural pesticide use, etc. So these bats are highly impacted by white nose syndrome. They are found mostly in caves and mines throughout the winter, and then during the summer they actually sometimes switch to using trees. So they use trees during the summer, so they're highly dependent on forested areas. On the left, this is the range of the tricolored bat. Now it's important to note that it doesn't hibernate everywhere in its range, specifically in that southern edge of it, because again, temperatures don't require hibernation there. So you can see there is a huge overlap between this used to be a very common bat and the fungus that causes white nose syndrome. To give you some context, where I work in Tennessee, we used to see tricolored bats pretty frequently. The area that we primarily trap in over the summer had the highest population of tricolored bats in the state, and we have only caught one in the last 3 years alone. So these populations are crashing. And so within this range, tricolored bats will primarily hibernate in caves. They're also being found to hibernate in mines and tunnels. And they're also being, also use these highway culverts. And so there are different characteristics for each of these things. So caves, you think about these caves are usually a lot larger. They can hold these very large colonies of bats. We're talking hundreds of thousands of bats in these caves. When you get deep into subterranean environments, that temperature is usually consistent and it's usually very, very moist. So it's a great growing place for this fungus that causes white nose syndrome. Some of the work that I've done previously in Montana, we put out some data loggers and we saw that this cave in Montana was all year round, no matter how warm or cold it was externally, it was 4.86 degrees Celsius 100% of the time. So these are really stable environments. Mines and tunnels are as well. Deep tunnels or deep mines, abandoned mines that go into that subterranean environment are going to again allow for that stable microclimates because they're not exposed to the external environment. So we do see a lot of bats use mines, especially out west where those cave systems and karst systems maybe are not as common as they are in the eastern US. And then recently we're seeing bats such as tricolored bats use highway culverts. And highway culverts are pretty interesting because most of the time they're not technically subterranean. Yes, they go below highways, but they're not actually in the subterranean environment. And they also are usually open on both ends and are pretty short. So it allows for a lot more flexibility of variation in that microclimate, a lot more wind flow that comes through. It's not going to be as different compared to that external environment. So this is a really unique system that we have here in Texas that we've been focusing on. Mainly, we're looking at culverts in this region here that's highlighted where the fungus that causes white nose syndrome has been recorded, but we haven't actually seen any white nose syndrome in these areas. Compared to caves not that far away that I'm pointing to in pink, do have white nose syndrome for this exact same species. So the main driver of this work was, why are these bats not succumbing to white nose syndrome? And there were two different hypotheses that we developed. The first was it's all driven by microclimate. These culverts are not going to be as stable. They're going to be, again, more exposed to the external environments. Perhaps it's just not a really good environment for this fungus to grow enough to cause white nose syndrome. The other hypothesis is perhaps these bats aren't actually going into hibernation, so their body temperatures aren't going to be at those environments that fungus needs to grow on. And so we did that first work looking at microclimate, and I'm just going to very summarize some of the work that we found because today's talk is going to primarily focus on the behavioral aspect. And so this is the work that my grad student Leah focused on for her thesis, and we just found out like 2 days ago, Monday morning, great email to wake up to, that her paper was accepted for publication by the Journal of Wildlife Diseases. So this will be out shortly for all of you to read. And so just to kind of orient you on this figure, we have hibernaculate temperature on the x-axis, and we have predicted fungal growth, specifically area, on the y-axis. And each of these colors are representing different hibernaculate types in the southern range of the hibernation range of this bat, of tricolored bats. So in black, we have cave data from Texas caves, those caves where white nose syndrome is occurring and causing declines of bats. We also have data from caves in Oklahoma, which also have white nose syndrome. We have data from tunnels in green. These tunnels are in South Carolina, pretty deep abandoned tunnels that don't, again, have that exposure to multiple ends. And these bats are also succumbing to white nose syndrome. And then we have our highway culverts in yellow. Um, what these dashed lines mean is the vertical dashed lines is, again, that temperature range of that fungus. And this is specifically that optimal range where that fungus grows really well between 12.5 and 15.8 degrees Celsius. This horizontal line is what we had essentially just determined was a threshold of fungal growth to cause white nose syndrome. And this is from lab studies where they inoculated bats with the fungus to see how much, hibernation can be shifted by the fungus. And what we found is that, as you can see in the bottom right of this graph, our culverts are too warm to cause significant fungal growth for these environments. So right away we can see it makes sense that even if the bats are hibernating, if they're matching body temperatures, their body temperature is going to be too warm for this fungus to grow significantly to cause any sort of white nose syndrome symptoms. We can also look at humidity. Again, funguses need water. They don't really grow well in dry environments. So we looked at the impact of humidity on fungal growth. And again, the x-axis here is relative humidity. The y-axis is growth of the fungus. These vertical lines is the humidity range of the fungus. Now, I just want to note, we have not studied humidity as much as we have temperature on the fungus. So we have a very clear understanding of how the temperature influences growth. We don't have that much of an understanding of how humidity or how much water is in the air influences growth. My lab is actually trying to measure this right now in the lab. But what we can say is that we have at least seen the fungus to grow at least in areas that are 85% relative humidity or higher. And what we found is that most of these culverts are also too dry for the growth to occur. So even if the air temperature does sometimes reach into that growth range, we're just not seeing it going to be in the combination of relative humidity and temperature to allow significant growth that causes white nose syndrome. Again, this work is going to be published, so if you're really excited about it, just keep an eye out for the paper in the Journal of Wildlife Diseases. So now I'm going to shift gears into that second hypothesis, right? Is it actually the behavior that's also allowing these bats to maybe not succumb to this disease? So there's a few different things we were interested in looking at. The first was, what is that relationship between air temperature of the culvert and the bat's skin temperature? So are they going into torpor? Are they dropping their body temperature to be at air temperature to reduce that energetic constraint of winter? Do we see that a nice relationship between that body temperature, which again, we're measuring a skin temperature here, and the external environment? If there isn't a relationship, is there a relationship between that difference between skin temperature and air temperature? Meaning, are they maybe dropping it to 5 degrees above air temperature? Is there a consistent difference between this potential mismatch? And the second piece was looking at how these bats are using these culverts. And this was driven by work that Sam has actually done looking at some of these bats are active during the winter and they are potentially leaving. So we're curious in seeing, okay, do they come back to the same culverts every time? When they use a culvert versus they aren't using a culvert, if they're outside of the culvert, is there a difference there in microclimate? And then how, if they're spending time in a culvert, how does microclimate influence how long they're in a culvert and that probability of leaving? So as I mentioned, this was a very field intense project. It wasn't a long period of time, but it was just a very intensive work that we did. And it was one of those projects that almost everything that could go wrong went wrong, except for the one thing I was expecting to go wrong. So it definitely taught us, and it taught our graduate student how to be resilient when you're doing field work. So we first deployed microclimate sensors, these kestrel drops that measured air temperature and relative humidity. And we put these in the central point of all the culverts in this area that bats were using. And we also put one on the external environment to measure that outside temperature and humidity. We then use pit tags, which are passive integrative transponders, and we put those into 350 bats of all of the culverts in the area. Essentially, these are subdermal tags that you can just scan each bat to see how they've been pit tagged and what their identification is. It's the same thing you'd put in your pet if you pit tag your cat or you pit tag your dog. And then TPW went back monthly to look at where these bats were and to scan almost every bat that they saw in these culverts. And then to get at this actual hibernation behavior, specifically with that body temperature, we use temperature sensitive radio telemetry. So basically we put on these tiny little tags, they're about 0.25 grams, and we use surgical glue to glue them on the back of these animals. It disintegrates over time, so these tags will naturally fall off. And what we do is we place receivers and antennas in the culverts, and it records the pulse rate of this tag. And what's really cool about these tags that pulse rate is actually proportional to temperature. So the receiver does some really cool math calibration, and it will actually give you the temperature to temperature recording of each of these bats over time. We did this for a month in January, which is about mid hibernation for these animals, and we tagged about 93 bats in 3 different culverts. These are the culverts that had the most bats throughout the ones that are in the area. And just to give you some context, we weren't able to permanently put anything into these culverts, so we had to be really creative. Something else to think about, a little tag that's sending out a radio signal, you have to think about it bounces off of things like a culvert. It might not make it all the way to the end of a receiver. So we had to put out antennas throughout each of these culverts, figure out a way to keep them off the ground because it does flood a little bit in there, to not have it be permanent, and to have them all be connected to the receivers that were kept outside of the culverts. So we put, I think I calculated it, over 2 tons of equipment into these culverts that we physically carried ourselves, and about 2 kilometers of cable of 36 antennas. So pretty intensive, but this part was the part that worked. I was convinced it wasn't going to work because we have done it before and we haven't had luck. So this part is pretty exciting that we got it to work, and we're hoping to be able to do this same sort of study in other parts of the state and see if we can get some really cool data. Okay, so let's dive into first what the skin temperatures look like. So if you're not someone who plays around with data and looks at things like data distributions, I'll kind of reorient you here. So this top figure here is looking at just what the culvert air temperature looks like. We have culvert air temperature on the x-axis, and on the y, it's just the proportion of the total data. So you can see overall, it's pretty variable. Most of the time it was spent between 15 and 20 degrees Celsius, but you know, it was definitely pretty variable in what those air temperature were. At the bottom figure, though, is the actual distribution of that skin temperature of those bats. As you can see, it was not as variable. They, had their skin temperature around 10 to 25 degrees, but they were not there, or they didn't drop their temperatures below about 10 degrees Celsius. So this either means they kept their body temperatures warmer when it dropped below 10 degrees Celsius, or they physically weren't there for us to record those body temperatures. And so here's an example of what these traces look like just for a couple days. Blue is the culvert air temperature. And as you can see, it's very almost consistent across these 3 culverts. They weren't that far away from each other. And this is just showing you that the culvert air temperature is not going to be that different compared to the external environment. Unlike caves, where it's pretty stable in a lot of areas, it's very variable here because it's driven by that external environment. And these black dots are the temperature traces of the bats, of all the bats in that culvert. So you can see there are periods of time where we're not measuring their body temperature, which we can say means they probably weren't in that culvert. And I'll talk more on that in a little bit. So the first question, which was looking at what is that relationship between skin temperature and culvert microclimate, we found that there wasn't one. Bats rarely matched that culvert climate. As you can see in the left figure, which is temperature, you would expect to see a really nice correlation between skin temperature on the y-axis and air temperature on the x-axis, and we see none at all. 80, almost 80% of the time, these bats were maintaining warmer temperatures than what we expect if they were truly in torpor. So they're really not actively hibernating in these areas. There also was not a relationship between the water vapor pressure, which is essentially a more scientific way to represent relative humidity, which with higher water vapor pressure, meaning there's more humidity in the air. We really didn't see any sort of relationship there. We did see a little bit of a relationship, however, with that difference. So this is the difference between skin temperature and the environmental temperature inside the culvert. And what we see here is that bats were, keeping their body temperatures a lot higher than air temperature when air temperatures got pretty low. And then they tried to actually be in torpor when the air temperatures were pretty warm, which is pretty exciting and different than what we expect. We also saw some really cool patterns with water vapor pressure. So when water vapor pressure was high, meaning it was high relative humidity, the bats didn't really seem to go into torpor. But as it got cooler and as it got drier, they did seem to go into torpor, which is pretty interesting. And they maintain those body temperatures, though, a little high. The second question was, did bats have high fidelity? What this means is, are they going back to the same culverts? Now, this is very hard to show at visualization. So this is a weird figure. I couldn't figure out a better way of showing it to you. Essentially, all the rows are the 3 culverts that at least we were focusing on for the telemetry part of this project. And all the columns are individual bat identification numbers. And those numbers are just driven by the radio frequency of those tags. Now remember, we tagged 350 bats. I'm just showing you the bats that we also tagged with those telemetry tags so that we can see, are these bats leaving the culvert and going to a different culvert? Because that was a worry for us because we were recording them in those culverts. So we were afraid that if they went to a different culvert, we wouldn't be recording their skin temperature. So it meant that they were potentially still doing hibernation behavior, but in a different culvert. What we found was about 96% of the bats stayed within the same exact culvert where we found them. So you can see there's only 3 bats out of these 90 so bats that went to a different culvert. And each time they only went like once and then they came back to that original culvert. This means that the data we received is that if we don't record a bat's skin temperature, that means that bat was not actually in a culvert at all. It actually means it's probably using trees, which is a pretty cool behavior to see during the winter because most of these bats are going to be hibernating in like caves and other types of hibernaculum. But in the winter in this area, they're actually using trees, which means they're probably more active and maybe they're even foraging, which is pretty cool. And then the second piece was, again, looking at use of these culverts. So this is a little bit of a confusing figure, so let me walk you through it. First part is the blue colors essentially mean these are summaries of environmental temperature, whether it's the culvert temperature or the external temperature outside of the culvert, when the bats were in the culverts. And then the orange is the mean temperatures of those 2 areas when the bats were not in the culverts. What we're actually seeing is that bats use these culverts when the culverts were warmer. So as culverts or environmental temperatures decreased, bats were not found in the culverts, which is pretty cool and pretty different and counterintuitive to what we'd expect to see. We didn't see any relationships with humidity. I know statistically it might look like it is, but those are just standard error bars. So if you look at the stats, there isn't any significant difference between humidity or water vapor pressure like we see in temperature. So temperature is really the important piece here. And then if we look at how much time they're spending in these culverts every time they go into a culvert, it's, again, counterintuitive to what we'd expect to see. We would expect to see bats using culverts to hibernate when it's cold outside, but it's not at all what we're seeing. we're seeing bats using these culverts longer when it's warm outside. So we looked at a bunch of different microclimate variables statistically to see what's the most important, and maximum external temperature during that time that that bat is in that culvert is what's driving how much time they spend in that culvert. Essentially saying that the warmer it is, the more likely that animal is going to be in this culvert rather than being outside in a tree doing some active behaviors. We can also look at the probability of leaving these culverts. So we have, now this is mean hourly external temperature. So this model is just essentially looking at every hour, what's the probability of a bat leaving a culvert. And we found that again, external environment is what's driving that probability. And as it gets warmer outside, the likelihood of these bats leaving is decreasing. So bats aren't actually using these culverts because they're warmer than the external environment, but they're using them as a refuge from warm external temperatures. Because again, we're at that southern end of that range where we'd expect them to be hibernating. So what's actually happening here is that these bats are pretty active. There's food outside for them to forage. But when it's warm out and perhaps they even go into heat stress, they're going to go into these cooler culverts where there is a nice wind tunnel and go into very shallow torpor to reduce that energy expenditure. So in general, we found that again, there was really strong site fidelity. Over 96 of the bats remained in the same culvert that they were originally caught in, which does not surprise me because some of the work that I've done previously where you go into these large caves and we can actually mark with Sharpie like where these bats are physically on a cave. And every year you can see this was 2021, this is 2022. Not only are they in the same areas of the cave, but they go into the exact same little spot. So these bats in general have very high site fidelity, but it is very exciting for us to say, okay, well, that means we can rely on the data we have, that if they weren't recorded, it means they were physically not in a culvert, but somewhere else. We do see that they do some partial thermoregulation where they do some shallow torpor. They will drop their body temperature to be lower, but they will not go into deep torpor, which means they're not going to match that environmental temperature. They're going to reduce their temperature to reduce that energy expenditure. It's just they're not going to go into this very deep torpor because they don't have to. And we also found that those external temperatures were really the strongest predictor for us to understand how much time these bats are using the culvert, what's the probability of it leaving the culvert, and when they are or not using that culvert. And so as someone who studies environmental physiology, this is really cool because it's always really interesting to see how behavior and thermoregulations specifically are different on the edges of an animal's range, because the edges of the range, they're going to experience different environmental conditions than compared to the rest of their range. It also means that it's very possible that it's a combination of one, these culverts are not conducive to fungal growth, but also the fact that these bats are not dropping into low enough temperatures for the fungus to even grow on these bats. So even if the culverts were getting cool enough and were staying moist enough for the fungus to grow, the bats themselves are not getting cool enough. And since that fungus grows in the bat, it just wouldn't be able to grow. So it is really cool to see this plasticity in how these animals hibernate, but it is something to keep in mind with climate change and the fact that warm temperatures are driving how these animals are using these artificial habitats. And we have to kind of think about that when we're looking at, say, management implications. So these East Texas culverts are pretty critical to this population of tricolored bats. They're a really good habitat for these bats, and again, not conducive to fungal growth. So it's a refuge from not just the environmental conditions, but also white nose syndrome. And so going forward, it's difficult to kind of think about. Normally, we want to think about the conservation implications to, say, changing a forest landscape, but to think about the conservation implications of changing an artificial habitat like culverts is important going forward, that we have to consider bats if we're changing anything about culverts in general. So with that, I will end the talk. I have my email there. I'm always happy to answer any questions that we don't get to today. I've also put Nate's email up there as well as Sam. They can also potentially answer emails about different parts of this project. I'm also, just to kind of put it out there, I do mentor students, and I'm always excited to talk to high schoolers that are interested in going into a career about wildlife, about what that might look like. Because when I was a high schooler, I had no idea that I could even do this. I wanted to do this. I did not know that this existed until I went to my undergraduate. So I'm always happy to talk about these things with people if they have the necessary need for it.
Thank you so much, Katie. It's fantastic to see the culmination of this very challenging fieldwork that we experienced.
Yeah.
Okay, if anyone has questions, please go ahead and write them in the chat. We've got a couple in there right now. So Mike McGrath said, regarding humidity, are the culverts used in this study primarily used as drainage, or do they convey a water body like a stream?
They're mainly used for drainage. So there is water in some of the areas, but it's not a stream component. So it's not going to be consistently with water. That flow is not consistent, but after a big rainfall, there will be some wash away in that. And I do believe, I don't know if it was Sam or if it was Leah who did see a fish in one of them. So, but it's not for streams. It's just runoff.
I will say I did see a beaver in one once. Robert Slaughter asks, are the tricolored bats not migratory?
These ones are not. So we have in general a variety of winter behaviors with bats. There are some migratory bats out there, but tricolored bats are not the ones. They're pretty small, so that usually is driving some of that. Our migratory bats that we have in the US are the larger bats that we see. But tricolored bats are pretty tiny. They're going to be shorter than your thumb, and like I said, weigh about a quarter.
Question from Vanita Fuller. She said, regarding the chart showing culvert location by bat ID, oh, got to scroll back up there. Do you know why some bats were in culvert number 9 on a regular basis and then moved to number 16?
No idea. I don't know why they all moved to 16. I can tell you that all three of these culverts were pretty close together. It's just possible that, 9, 16, 8, and they would hit 16 first and decide to go there. Most of the culverts that we worked in were pretty similar in terms of length, pretty similar in terms of size. And some of them had more bats than others, but in general, they were fairly close in terms of the bats that were there. So not sure, because we didn't see a lot of movement. I think if there was a lot of movement between culverts, we could probably pry apart why they'd go from one culvert to the other and back. But since most of them stayed in the same culverts, those rare instances, we're not sure what's driving that.
Okay, and Wendy Anderson asks, was there a maximum temperature the bats tolerated in the culverts?
I didn't see anything in particular. No, granted, this is over winter, so this was January, so I don't think it got above 25 degrees Celsius. I do think it would be interesting to see as it got warmer into, say, March, what that would have looked like. But Sam can correct me if I'm wrong, but by March or April, they're not using the culverts that much anymore. So they're not going to maybe see and experience those extreme temperatures. But in terms of was there a threshold? We did not see one, but I don't think that doesn't mean there wasn't one. It's just, I think it was just because of what the time of year that we looked at this information.
Yeah, I can confirm that by sort of March time, there's maybe a quarter of the bats still remaining. And then over the course of the summer, there's, 1 or 2, usually males that hang around. Okay, Leland Hefner asks, does the fungus die when temperatures are above 19 Celsius?
No. So if you I'm trying to see. Essentially, what happens outside of those temperatures is it just stops growing. So the fungus can persist in substrate. It can persist in the cave without bats there. That's why it's so prolific because it can hang around all summer. But it just won't grow without something to feed on, and it feeds on the proteins and skin of the bat wings and those. Um, there will be a point that it will die. It just will not grow, and it can't persist as much beyond that 19 degrees. Now, I do want to say that that work was done back in 2012 in the lab. There has been very recently, like within the last couple of months, work that has been published that shows the fungus is growing in higher environments as high as 27 degrees Celsius. Um, we know that there isn't a lot of genetic variation within the fungus, so it potentially could be a local adaptation of the fungus, but it's more than likely that it's hard to replicate what we see in the wild in a lab. You can only do so much. It's very difficult to grow this fungus in the lab as I'm learning. We try to grow it on, say, a bat wing compared to like your classic medium. Um, and so things might be a little different in the wild when it's on a live bat wing versus the lovely potato agar that we have in our labs. So we know it doesn't die at 19 because we know it can actually still grow a little bit higher. So I don't think we still know yet what that actual true limit is of that fungus.
Thank you, Katie. We have a question from Grace here, complimenting your wonderful presentation. She mentions that they are also on the edge of the range in Nebraska, and that tricolored bats use culverts in the summer. She says she sees a lot of conversion from bridges and box culverts to corrugated metal pipe culverts. Do you think suitable culverts, roost trees, or white nose is the most limiting?
So I would say that it's probably a combination of all the things. So like I said, these bats are tree roosters in the summer most of the time. And we found, at least in Tennessee, they tend to like just hardwood species, but that can be very different across their range. I think depending on where they are, it could very much be the habitat and the suitability of just persisting. One, do they have habitat to have enough food? That's going to drive if they can survive white nose, if they have enough fat to get through white nose. We know now that fat going into hibernation is huge for survival of white nose because it can allow for those that increased arousal rate. If it has good suitable habitat for winter that it could persist in and keep its energy down, that's also very important. And then three, of course, white nose syndrome is going to be a driver. So I think habitat is more of can really modulate that effect of white nose. So I would say habitat is key, and someone who always thinks about thermal biology, I would definitely say habitat is key, because there's also been a lot of work out there, because we know fat's important and we know microclimate's important. People have been doing research and saying, can we adjust mines or tunnels or culverts, your man-made things that they hibernate in? Can we change the microclimate in these environments to be not very good for the fungal growth, and actually reduces energy expenditure of these bats? So I would say habitat would be at least my most important thing, mainly because we can't really control white nose, but we can potentially help them beat it.
Okay, we have 9 more minutes, so I'm just going to
Okay.
choose a couple more of the questions that we have in here. So Stephen Vanderkamp and Lewis said, I know you looked at bats in East Texas culverts, but do you think they would use culverts in Central Texas for similar reasons, i.e. heat refuge? Any thoughts on reason for cave use during winter, also for heat refuge?
So I think, it's one of those things that I know that Nate, our collaborator, has talked about why they use culverts in some areas, and we're not always 100% sure why they're using a culvert versus something else. Is it an additional habitat that they didn't have before, or is it because we've changed their original habitat and they need another area? So I'm not sure if they would use. I feel like if they didn't have, I think caves are probably more of the priority that they would use. But Like, I don't know if like putting culverts out there for a refuge would make a difference or not. Um, Not 100. I can't answer that question, I guess, to give it the short answer. I could hypothesize all day, though.
I can say that TPWD have funded quite a lot of research doing culvert surveys for tricolored bats across the state of Texas, and very much so it seems to be this very short stretch where this research was done where we're finding those very high numbers. Otherwise, it tends to just kind of be a smattering here and there.
Yeah. Mm-hmm.
Okay, couple more here. Is supplemental feeding used within their range as a conservation measure to try and ensure tricolored bat survival in the winter?
So, in terms of exclusively for tricolored bats, I can't say anything about that. But I do know that Bat Conservation International has been doing what they call the Fat Bat Project, which I love the name of that, where they are looking to see if they put out light sources near hibernacula in like the summer and fall, when these bats are trying to stock up on that fat stores before hibernation. If it increases the foraging availability of insects, does it increase and result in it gained more fat, essentially providing that supplemental food? And then does that allow for more survival from white nose syndrome? So I know the project is still going on, and what at least they have published on is that they have shown that artificial light has increased insect abundance in these areas, and it also, I believe, has increased foraging behavior in these areas. I have not seen if they've published any work yet to see if that results actually in higher fat stores, and therefore if that has resulted in higher survival from white nose syndrome. I do know that fat is a huge driver of survival, and they've shown this in a variety of species. And so I can assume that if they are increasing how much fat they have going into winter, if they can get it to a point that it allows them to withstand those increased arousals, then supplemental feeding through just artificial light could be pretty neat in a survival way.
Thank you. So we also have a comment from Diane McMahon. She said, I've often wondered if Mexican free-tailed bats are a vector for spreading white nose syndrome. Can you talk to anything about use of the culverts by Mexican free-tailed bats at all?
So I don't know in terms of Chiderda in the culverts, but I know that there has been work recently that has shown that the fungus has been found in southern Mexico. And so the assumption is that a migratory bat such as Chiderda that overlaps with areas that has the fungus has picked it up and has brought it there. And we're trying to submit this paper like right now, looking at least the microclimate suitability of some of these Mexican caves for the fungus. And there are areas in high elevation in at least central Mexico that are absolutely conducive to fungal growth. So it is definitely a possibility that if the fungal spores can hitch a ride and survive that ride, which is pretty crazy to think about in terms of that's a very high body temperature of that bat holding, when it's flying. But if it can get there, it could potentially be a problem. Now, it's important to note that just because that fungus is there doesn't mean the bats are hibernating there. And that's also a key, right? Bats have to hibernate long enough for it to be an issue. We do know bats hibernate in these areas, so there is a potential, absolutely. Will it happen? We're still gonna have to wait and see.
Yeah, all very, very good points. I'm just going to keep going until we run out of time, if you're okay with that, Katie.
Yep, sounds good.
Okay, so Barrett Clark says, do the results of the study indicate any potential implications for or insight into culverts as a function of disease, i.e. white nose syndrome vector sites?
I do think in terms of like vector sites, I do know in other parts of other areas where tricolors use culverts, they're getting hit by white nose pretty bad in like Georgia and other areas. So I think it was just the Goldilocks effect of it just being in an area that had the right microclimate that didn't allow for the fungus to grow, as well as it was on that just edge enough that they're not completely going into deep hibernation, I do think looking at culverts exclusively is not the way to go, but rather thinking about the behavior element, because culverts in other areas could potentially absolutely provide growth of the fungus. And if the bats are hibernating in those areas, then a culvert wouldn't necessarily be a reduction in the probability of white nose, but it could absolutely be an area where they will have white nose. And then if they do move to other areas, then you could actually see that spread.
Okay, one last question for you.
Okay.
From James Raymer. So roosting in culverts, either in torpor or heat refuge, significantly increases long-term survival. Question. Do they need crevices or are smooth surfaces suitable?
So great question. So we didn't actually look at survival from white nose, right? We know that they are surviving. They don't even develop white nose. So they are surviving the fungus. So we can assume that there is survival in these areas. Whether it's due to the microclimate or the behavior or the both, we don't know. It would be really cool to see, okay, can we measure microclimate in areas where the bats are going into full torpor for full hibernation? Can we measure the microclimate and model the fungal growth in areas where we'd expect the fungus to do really well, but the bats are not going into full hibernation, right? So we can't untangle those two things based on the fact that they're both happening. If we could control, everything in the environment, I would love to do a study where you can say, okay, this microclimate should have growth. The bats aren't doing hibernation. This microclimate shouldn't have growth and the bats are doing hibernation. So you can untangle, is it a microclimate effect or a behavioral effect? So that at least answers your first question, that we think so for increasing survival. And then in terms of the crevices are smooth surfaces, that's actually a question that we have had for a while. We just haven't figured out how best to ask it, because in the culverts, just through personal experience, and Sam can definitely dive into this because she's been there a lot more time than I have, these bats do tend to like those seams of the culvert. So we would think that, is it because that seam provides some change in microclimate, or is it just easier to grip? Or does it provide, and we saw this a couple of times, like, does the water come to, just due to like diffusion of water and how water works, it tends to kind of go along those seams. Maybe they like the fact that they're just getting water coming to them. We don't know. We think that crevices are important just because of where we see the bats, but we haven't been able to actually test that yet. Again, it would be something that we'd have to find a culvert that is just completely smooth and see how the bats are different between them. And that's probably never going to be something that we can find unless we build it ourselves.
Thank you. So
Thank
we
you.
are at the 1:30 hour. So thank you again, Dr. Hass, for your fantastic presentation. I'm sure lots of people here are excited to read the paper. Again, Dr. Hass's email there, along with Nate Fuller and myself, who are all involved with the project as well. So I'd like to thank all of you for joining us for the seminar today and supporting the research that we're doing.
Great. Thank you so much for inviting me. This was awesome.
You are very welcome. Everyone have a great day.
Bye.
Bye-bye.